So welcome everybody to the September 8th mini seminar of the IMAG MSM Working Group on Multiscale Modeling and Viral Pandemics. Although our seminars are span a little bit beyond the scope into the immune system and multiscale modeling in general. This seminar is being organized, co-organized by myself and James Glazier, and then Jim Sluka helping with the web administration, Slack, and communication in general, and of course Lorenzo and Joel, who have been instrumental in getting invitations to speakers out and ensuring that we have speakers for as many Thursdays as possible. Please note that we have different communication channels, including Slack, Twitter, wiki page, and LinkedIn group, as well as YouTube group or YouTube channel, where these seminars are being live streamlined and also archived, so you can see them. If you miss a seminar, you can see them there. Please follow us on Twitter, and as we make announcements, help us spread the awareness of this group as well as the seminars. We have a couple of announcements today. So NIH is launching this L3C or Long COVID Computational Challenge, where they will be awarding up to $500,000 to develop data-driven or machine learning driven solutions to be able to predict the likelihood of patients developing long COVID symptoms. There is a link here that you can follow if you want to, for yourself, if you want to collaborate with others in the group on applying for this. Might be a good opportunity to check out. Another announcement is that so our university at the University of Nebraska has launched a Grand Challenge competition where over 4 years they are investing $40 million total, so $10 million each year into big idea projects. And so this year was the first year of this competition, and humbly so we were lucky, and we received a $5 million grant and support from our university to build a digital twin innovation hub, which will start developing multi-scale infrastructure for a digital twin of the human immune system. I'm happy to, if anybody's interested in this, I'm happy to collaborate and work with the community as this project gets underway. And third announcement is a paper from our group on compositional modeling of immune response and virus transmission dynamics that you can check out here as well. Any other announcements from the community? Okay. If the community has any announcements, please feel free to email them to us, and we'd be happy to include them during these seminars. And any announcements regarding additional meetings or funding opportunities or funding obtained that's relevant to the group, publications or requests for help or collaboration, please let us know, and we'd be happy to include them in the announcement section of the seminar. Upcoming seminar on September 15th will be by Herbert Sauro on dynamics and sensitivity of signaling pathways. September 22nd and 29th are not filled yet, so please feel free to, we always appreciate suggestions for speakers. Please let us know. We have a Google form here that you can also fill out to suggest speakers, but if it's easier for you to just simply email them to us, please do that as well. Really, any help is greatly appreciated in this area. So without further ado, it's my pleasure to introduce Dr. Paul Thomas from University of Southern California, who is our speaker today, and he will talk about causal activity modeling framework and application to viral and host response processes. Thank you so much for your time, and we look forward to your presentation.
Thank you. All right, let me try to share my screen here or share my presentation. All right, can people see this now?
Yes, in the presentation. Yep.
Presentation mode.
Presentation. It's you've got the 2 slides there with the notes and stuff. Yeah.
Okay, that's unfortunate. Okay, hang on.
I think if you click swap displays, it probably will change right there on top. Yep.
Ah, there you go. Thank you. All right, how's that now?
Perfect.
Good.
Great. All right. Thanks. All right. So thanks again for the invitation. Yeah, thanks Tom and Lorenzo and Jim for inviting me to speak here today. So I'm going to talk a little bit about the gene ontology causal activity modeling framework. This is called, we call this GoCam for short. And then I'll touch a little bit on the viral and host response processes, but really this is not my area of expertise, but I can at least show you the work that our consortium has done so far. So, you know, I'm a professor at the University of Southern California in Los Angeles. I'm also one of the PIs of the Gene Ontology Consortium. The Gene Ontology Consortium is really large. We have a lot of members. And so I'm representing a really large project today. So for the talk, I think I'm just going to briefly go through an introduction to Go and GoCam. Talk a little bit about the Noctua tool that we've developed for curating GoCam models. So basically for making causal models of processes. I'll at least show an example of one of the GoCams we have for a viral process. And this is really this is going to be, it's going to be a model of SARS-CoV-2 and interactions with the human immune response. And also, it's probably towards the end of the talk, I'll walk you through the available GoCams we have in this area of biology. And then, I'd love to afterwards, obviously, discuss potential collaborations opportunities, what we can do to help you and maybe vice versa. So, okay, so this, all right, looks like the animation didn't work great. So what I'll start with basically is for people who are not very familiar with the Gene Ontology Consortium and our products, the Go is really, the premier source of function information about genes. And this is genes in, in any organism, including viruses. And so it's really it's designed to be a representation, as complete a representation as possible of current scientific knowledge about gene function. And by this, I mean, the representation is both human understandable. And so all the definitions, for example, of gene ontology terms have human readable definitions. But also it's, and I think this is really the most important application of the Go, is that it's computable, right? So it's represented in terms of an ontology that is, a formalization of really our current understanding of how genes function, and then annotations, which assign specific genes to functions. And so you can kind of think of the 2 main products of Go as, on the one hand, the ontology, and think of this really as a structured representation of any possible function, a way of describing the way that gene products function in systems. and it's made of classes, ontology classes, the relations between those classes, and definitions. We have both human readable definitions and machine readable, computable definitions that can be used in all sorts of, logic, based analyses and take advantage of the semantic web infrastructure. So, and then the other part of Go are statements about gene functions. Right. And we really have two formats for that. The sort of standard format that probably most people are familiar with are the annotations, where it basically associates a particular gene. Right. So I'm giving a couple examples here. A particular gene or gene product to a function. And we have three different axes of function. We call them aspects of function that we try to model in Go. And one of those is molecular function. What's the gene product? Usually a protein, doing it at the molecular level. So an example would be it's a glutamine tRNA ligase. We represent larger processes, biological programs, you can think of it, and that are carried out by multiple gene products and functions acting in concert. And then, of course, each function is carried out in a place in the cell. And so we have what we call the cellular component ontology that describes sort of cellular components and where gene products are active. And then, more recently, and this is why I'm here, is to talk about Go causal activity models. And you can think of it in a simple way as linking all of these, what we have traditionally had as separate statements in Go, as separate annotations, linking them into an overall model of a process. And so you can link the sort of basic annotations using, of course, then causal relations to describe how, say, one activity will act on another. Oops, I'm having trouble with my pointer here. And act in concert during a larger biological process. Okay, so the Go statements about function have always varied in their information content. So it's it partly depends on what's known, right? So a lot of times we don't know much about the biology. We may only know that gene product A binds to B, right? And you can sort of see now as we go down this list, I'm making more and more precise statements about the function of that gene. Right. So I'm now able to say that, well, A actually is a protein kinase that acts on B. And you can sort of see that eventually I can make statements like, A and B are involved in signaling to regulate the cell cycle. So things like this. So it goes, the gene ontology is designed to be extensible and to handle all of our sort of unknown, so basically handle whatever level of resolution at which we understand the biology. Okay, but of course, then it's also partly depends on how we represent it. And this is really where we were running into difficulties with our standard annotation paradigm is when you can only associate one gene with one Go term, you're really limited in the complexity of the biology and how well you can represent computationally a biological system and how it really works at the, as linking together molecular activities. And so that was the impetus behind the GoCam modeling framework that I'll talk about today. So just to make this a little more concrete, I'll give you an example. Okay, so this is a NED4 gene. So you can think of individual gene annotations, right? In standard Go annotations might have been something like when you can make one statement about NED4 being ubiquitin protein transferase. Okay, so basically a ubiquitin ligase. Okay, I can say separately that it binds to RNA polymerase. Right. And then I can also say separately that it's found in the nucleus. Okay. So I can make these individual statements. And of course, the idea is then, obviously the full model of the system, you have to be able to link all of this information into a description of how it all relates to each other. So in GoCam, you can actually make the much more precise model by linking together all these individual pieces of information, right? That basically NED4 acts as a ubiquitin protein transferase. It acts on a particular substrate when it's in a particular location as part of a particular biological program. In this case, it's the cellular response to ultraviolet damage. right? Damage from UV light. And it acts directly on RNA polymerase, which itself has RNA polymerase activity. This is a GO term, and it occurs in a place. So you can start to actually model this, and we then have these causal relations, right, that it negatively regulates it. So the idea, you can see, is to go beyond these sort of isolated annotations and statements to a more rich model. And this was something we published a couple of years ago in Nature Genetics. So if you haven't read this paper, it's short, so you can probably get through it pretty easily. And the real idea here is that it's a fully computable model. It's based on OWL instance graphs. So this is ontology web language. This is sort of really the standard for semantic web representations. All of the elements of the model utilize ontologies, meaning it's all computable with ontologies. Obviously we use Go for functions, cellular locations, biological programs or processes. We use the relations ontology for the relations, the cell ontology, and Uberon. So Uberon is an ontology that describes tissue and tissue types and anatomical features, and then the Kebi ontology for chemicals. So everything in the model is computable. And then just like Go, it stores all of the evidence for each triple you can think of in the model, right? Each linking up of two ontology objects by some sort of relation. Okay, so this is the core data model. I'm not going to go into a lot of detail, but if you have questions about this later on, we can go into this in nauseating detail. But I will just give you a general flavor for what's in this model. The core of the model is, what is a gene product doing at the molecular level? This is its molecular activity, and it's represented by Go molecular function terms. Okay, and these are what are going to be linked. One molecule acts on another and causes some change in its activity. So that's the basic, molecular biology modeling paradigm, right? Which is how molecules influence the activity of other molecules. And then you can specify all sorts of additional information about that. So which molecule is it that's acting? What is it acting on? Where is it when it's acting? And of course, we can say at the cellular level, at the level of a cell type, or at the level of a particular anatomical feature, or even, which organism is it operating in? So you'll see that, obviously for modeling viral processes, we have to model gene products from more than one organism, right? So host virus interactions. And then the molecular activity is carried out as part of a biological program, right? And this is the Go biological process. And of course, these can be nested. You can have modules inside of larger programs. And then, of course, you can specify that it happens at a particular temporal phase. So this might be a developmental stage. This might be, a stage in an illness, whatever things that we want to specify temporally. Okay. And then, as I said, we have a detailed evidence model that stores all the information about, who was it who put this statement in the database. So we have full credit to anyone who makes contributions. What group were they part of? And this is really a way, operationally to acknowledge funding. Then there's obviously a date at which it was put into the database. What is the evidence, what is the evidence traceable to? Usually a publication. And then, we have ways to characterize the type of evidence it is. And then this sort of evidence goes on every triple, right? So you can think in this case, if we're making part of our model has a statement in it that says this particular molecular activity, let's just say protein kinase activity, is carried out by a particular molecule, let's just say, I don't know, MAP kinase. Okay. And so, let's say MAPK1, whatever. And so it is a specific gene product. Again, it's fully referenceable to a computer identifiable object. It's not just a string. And then that triple then has evidence that supports it. Okay. So each triple in the model can be built from evidence from different sources. Okay, and that's all traceable and and attributable to the people who did all the work. Okay, so I'll just give you a flavor of one of the pathways. So this is our pathway. It's called SARS-CoV-2 host. It was created by Patrick Masson, and actually he's the person who's made most of the models, I think, in the area of biology that this group would be interested in. And he's in the Uniprot Swiss Prot group. So again, all of this is captured as part of all of his models. And this just gives you, we have a sort of high level pathway view where you can get an idea of what's in the pathway, how, what sort of interactions are there, what sort of causal influences, and overall, what does the model look like? And again, I'm happy to have you guys look at these in detail, and we can talk about them, but just expect that my expertise in the biology is, I could always bring Patrick on a follow-up call if we want to have one. And so what I'll show you now is underneath the hood, what's here. And I don't know if you're able to read, can you read some of the text that's in these green boxes?
Not
Yeah.
a little bit, yeah.
Yeah, so it's what I'm trying to do is give you a little bit of flavor of the detail of the model. So what you really see here is I'm just representing each activity, right? Because it's really not, it's modeling what things are doing, not the things themselves, right? Obviously the things themselves are part of the model, but it's they're carrying out a process, so they're doing something. Okay, but of course, in a high-level overview, you're all familiar, right? A high-level overview of a pathway, we usually label these things with the gene product, right? That's carrying out that function, that activity. Okay, so that's why we're showing it here. But really, there's a lot more to the model than that, and that's what I'm going to try to show you. And so I wonder if I can. We can always go. I can always show you the tool later on live. So, but just to sort of walk you through this, right? So let's take this. So this is basically signaling through MAVs. So this is detection of, in this case, this is it shows essentially how the SARS-CoV-2 virus is trying to shut down the immune response by acting on human proteins in the MAVs pathway. Okay, and so here, for example, you can see. Oops, I'm having trouble with my pointer again. There we go. So we've got the N protein of SARS-CoV-2, and the model has all the richness of the gene ontology. Right, we're saying that actually its activity is protein sequestering activity. So it's actually sequestering its target. Okay, and it's acting on TRIM25, the human TRIM25 gene product, in the cytoplasm, and as part of we actually have a GO term that describes these biological programs that are enacted by viruses, and this particular one is suppression by the virus of a host type 1 interferon pathway. So that's basically, so these are all controlled, terms, from the gene ontology. And then it has a negatively regulates relation, right? So it's regulating the activity of TRIM25, which itself is has ubiquitin protein ligase activity, meant to, of course, when it's working properly and not interfered by the virus, acting on DDX58, okay, again in the cytoplasm. So just give you an idea of the richness of this model. And each statement in there can be based on different, evidence from different publications. Okay, so that'll give you an idea. And this is actually the curation tool. So I can just sort of give you a brief intro to how we make these models, or how curators make these models operationally. It's really just sort of a pathway building tool, where of course it's all linked to back-end software that makes a computational model about of this process as you build it, including, as I said, all the links to the evidence. So you drag and drop a new activity onto the map, and then you fill in information about what is the gene product, what's the activity it's performing, what's the evidence, where does it occur. And models can be incomplete, right? You may not enter evidence right away, or you may not know in which part of the cell in which it's operating. So you can make the models arbitrarily incomplete or complete, depending on your knowledge or what you have at hand. And then, of course, one of the big advantages of this computational representation is that it can be broken down, of course, into the standard Go annotations that it's made up of, and those can be computed over in simple Go analyses, in addition to all the sort of pathway-based analyses that one can perform on the richer pathway representation. And so just to give you an idea, again, I'm not going to go into detail here, but it's really that, of course, there on the back end, there's this rich RDF OWL representation of the complicated, basically the entire causal pathway. And it's done in terms of, technically it's done in terms of instances of ontology terms. So what that means is that everything in the model can be cross-referenced to to some object in an ontology and can be computed over in that ontology. So it's a very rich pathway representation. So that's really the extent of what I wanted to show you. But I'll go in after this. I'll show you a couple of actual examples as a way to kind of segue into a discussion. But really, the main point I want to get across here is that GoCam provides computable models of pathways and systems. And what I'll show you now, and we'll talk about a little bit, is that tens of GoCam models have already been created for viral and human immune response processes. And of course, I have to acknowledge the whole GO Consortium, because this really is a massive team effort across a really large consortium involving a lot of different groups and actually a lot of different funding sources. So while I want to make sure I point out the main funding source we have is a U24 grant from the National Human Genome Research Institute, NHGRI. That funding really only covers sort of the central functions of the consortium. And there are a lot of other grants that go out to individual GO Consortium groups. And you can read about all of the contributors actually and all the different consortium members at this URL here. So thanks for your time. I'll stop here, but then just in a moment I'll show you a little more detail about the model repository and some of the models. So thanks.
Thank you so much. Really appreciate it. Is it a good time to take questions, or did you want to go through the repositories now? Or?
Yeah, I thought it's kind of whatever you think is the most useful. If there are questions sort of about the modeling framework and details about how we make these GoCams, and I can answer that maybe first, and then if we want to get into specifics of models in the area of biology that you're interested in, maybe we can wait on those.
Okay. Any questions? I have a couple, but we'll take questions from others first.
I have hopefully a quick one.
Yeah.
So these are OWL constructs.
Yep.
So presumably you could use a protege or some other OWL editor as well.
Yeah, that's right. Yeah. And in fact, when we were early on, when we were prototyping all of this, we actually did it all in OWL. I mean, in protege. And we actually manage the ontology. All the Go ontology editing that we do is done in protege.
Okay. It just doesn't have the pretty diagram layout stuff.
Yeah, you can read in a model with protege, but it's really hard to interpret. And it's really, we obviously for curation and for most people to interact with the models, it's obviously we built this visual what we call the visual pathway editor to be able to build these models really easily. But yeah, it's all stored in the back end as OWL at the end of the day.
Hi, I also had a quick question. So some of these GoCam annotations, they are actually at the level of pathways, like one protein regulating the other. And that's, similar to many other existing resources like Reactome, for instance, aggregation resources like Pathway Command. So to what extent can you draw on those resources to fill in the pathway level information in these GoCams?
Yeah, so we actually have a so you may know that Reactome is a Go Consortium member. Yeah, and so obviously our first foray into this is conversions of Reactome into GoCam. So we actually have a separate grant to do that. It turns out to be fairly complicated to make a conversion between a what's really sort of thought and known as the process notation for pathways and the activity flow notation, which is really what GoCam is. So yeah, I know you're aware of this. Yeah, well, it's good to see you.
Yeah, we should follow up. But I had another.
Yeah, sure. But anyway, that's our main challenge. So bottom line is for metabolic pathways, the conversion is very straightforward. We have a full conversion. We actually already trade information and make sure that there's internal consistency between Reactome and these GoCams for metabolic pathways. And we're in the process of working out the more difficult problem of doing the same thing for signaling pathways.
Cool. Yeah, no, I think I understand what you mean about the difficulties. I had another question which is related to curation. So like, do you have in terms of the process of curation, how you prioritize which GoCams to curate? Do you have like a plan or something in place to how to prioritize that curation?
Yeah, so it's a great question. We basically, because the consortium is very wide, we actually have left the prioritization up to the contributing groups. Now we do have coordinating calls. So all the gene ontology curation is done in a very standardized fashion. So we have weekly meetings actually, among all the curating groups on sort of the standards. And that's also a chance for exchange about what each group is working on so that we would avoid duplication of work, right, among consortium members. That's our main way of managing it, is really, it's each group, depending on what they're funded to do, will have their own priorities. And then the central group basically just makes sure there's no duplication of effort and that everybody's adhering to the same standardized practices. So that's actually how we sort of do it. And so traditionally what we've done is, if there's an area that of biology and this is a perfect question because it's a great segue into what we might, what a possible model for an interaction with you guys here on this call, which is we've had collaborations in particular areas of biology. And what we will typically do is go out and find funding, or there may be existing funding in a particular area that we can then focus work on. And so one of our more recent ones has been on this synapse biology. So we had a group called SynGo. We actually published a pretty high profile paper on the work of that consortium. We've done in the past, we've done representations of apoptosis. And again, we get together the world's experts and try to hash out how all of that should be represented computationally in the knowledge base. And so this is kind of one way that we then address particular priorities. And so we really look to I think of the Go project is completely designed to be an open consortium. And so we can take in additional groups that want to make contributions in a particular area. And we really welcome that because obviously, even as large as the Go Consortium is, we don't have world's experts in every single area.
Thanks. That's great.
Yeah.
So I got a question related to contribution. So you track the author of any given contribution. Do you use the contributions in any way to weigh them? Like if it comes down to authorships and is there a consideration for significance of the contributions, or is somebody who will rename a component, their contribution will be weighted equally as somebody adding, a sub pathway or something like that?
Yeah, no, that's a great question. So actually, yeah, so as you saw from the evidence model, right? So it really is very detailed. So let me go back to this because this is a really important point. This is every single triple in the model. Okay, so it's every single little statement. So if I have a statement about, about a pathway, it will involve hundreds of separate statements. So credit is given individually for each triple, depending on the curator. And so what we tend to do is, what we do in our curation tool is we just display all curators. Right? Anybody who put at least one triple on the model because they at least did something for that. But yeah, you're absolutely right that we order it in terms of who's contributed the largest number of individual statements. So we really do track, so for example, it may be that, there's an existing model for a process. And so rather than recurating the entire process, it may make sense to then have a discussion. So what we encourage, of course, is this really is a collaborative process. So if there's somebody who's been made the overwhelming contributions to a particular model, we really like to encourage, anyone who wants to make changes to that model to contact that person. So we actually have a whole GitHub repository where we actually track requests for changes. And so, for example, even if you're not a GO Consortium member, you can start an issue on our GitHub tracker and say, hey, you're missing this interaction in this pathway. Can you add it? And you can say, hey, this is the publication I'd like you to use. I'd like you to credit me, and we can talk about how that might be done. So there are lots of ways in which to do this. Obviously one way is to give people curation access so they can actually curate models. Another one is just to interact with existing curators, just to make a request to go, and all of these sorts of things we can work out attribution models for.
Okay, and so related to this, the collaboration on the curation, is that can multiple curators work on the same, since it's web-based, can multiple curators work on the same model in real time like Google Docs, or
yeah, it's exactly like Google Docs. Yeah. Okay. Yeah, and we actually find that that's a really useful feature. Because you can real time watch what someone's doing. So you can be on a a Zoom call and cool. and sort of manage a collaborative project. Yeah, that's correct.
Other questions?
I have another quick one. Hopefully it's a quick one. You mentioned it early on. A lot of proteins have multiple functions, and so it's a problem to ascribe a single function to a protein unless you just start aggregating. There's a protein called ADAMTS1 or 1 through 9, a disintegrant and metalloproteinase with thrombospondin motifs. Yeah. So the name all by itself lists 3 different functions of the protein. So that has been circumvented in the GoCam structure.
Well, that's so Go has always been able to handle multiple statements about function. So a gene product has never been, because for exactly the reason you say, there are many proteins where you cannot describe all of its functions adequately with a single term. Right. And so, but typically what you can do is, you can break down its activity, in a pathway representation to what it's doing at a particular time and place in the cell and as part of a pathway. So that's why the model basically, encourages the use of a single Go term, but there's no reason you can't have a cascade of functions of an individual molecule that are carried out in a particular causal fashion. So basically, obviously in the model, we sometimes can break down those functions, and you want the detail of breaking them down into individual activities. But often, it's just that one activity is used in one context as part of one pathway, and another activity is used in another context as a part of another pathway. And that's exactly what this is designed to capture. Yeah. So if there aren't other questions, I can just go to the model repository if you want to just take a look at kind of what we have there now.
That would be great. Yeah.
Just give you a quick overview. So what I've done is I've gone to, so we're actually in the process of revamping our public interface for public-facing interface for GoCam. So it's a little easier actually just to show you our curation tool. And so you can get an idea of what's there. So I've already filtered it by the models you're likely to be interested in, okay, which are the ones done by Patrick, as I said. So if you just go to this website, you can go to noctua.geneontology.org. Okay, and you'll be able to do the same thing I've done here, which is just, add a contributor. And then I've also filtered his models by production. Okay, so that's the other thing that's really nice about the system is people can curate a model and decide on when they want to release it to be publicly visible. So by default, when you start a model, it's all in development status, and it's not shared with the outside world. It's not something we publish. But then if you add the production filter, you can actually once you've chosen to change the state to production, it then goes through the whole Go release process and is released publicly into the Go knowledge base. So these are and I'll try to make this a little bigger for you guys. Let's see if I can, whoops. Oh yeah, I'm in the wrong app here. Let's try this again. Okay, there we go. Okay, so now you can see maybe some of the titles of the models. Oops. Whoa. Okay. Yep. And so you can get an idea of the kinds of models that are in here. So there are 80 models that he's created. Most of these are so you can see basically the way he's tried to name these is the systems that are involved. So if you see human and some sort of pathogen, it's basically going to have gene products from both of those interacting. So it's basically in this case the interaction of human defense response with particular virus. And so you can kind of get an idea of the way these are named, and each of these are things that you can explore. One of the easier ways to get an overview is to go in here to this. Oops, sorry about that. I made everything too small. To the pathway preview, and so this sort of gives you the overall, the high level view of the pathway and what's in it. And you can mouse over any of these. I've sort of blown things up a little bit too much here. Maybe I can shrink this down again. You can get an idea of this tool. Basically, as I said, it gives you the high level view. But whenever you mouse over something, you can see that it has a little card on the right-hand side that gives you all the gory details on what that actually is representing. It's not just a label on a gene product. It actually tells you what the gene product is, what it's doing. In this case, it's cyclic GMP or AMP binding, where it's occurring. And so this basically will just sort of give you an idea of what's in each of these. So if you want to explore these further, so that'll give you just a quick look into what we've done in the specific area of biology that you're interested in. So yeah, we can scroll through these, but yeah, anything else you guys want me to look at? If there's one of these you want me to launch, or I don't know if you had a chance to read these.
How did you do that original filter? Did you just filter on something like virus or infection?
I filtered on
Or
that.
did-
So, yeah, so that's what's in our public facing. But as I said, we're revamping it right now, so it's clunky. And so this is, what I'll do is I'll send you guys the URL when we've got it released again. But what I've done here is I know who the curator is, who's made all the host defense responses and viral interaction pathways. And so I just did a search for Patrick. So you'll see. So if you can actually see the total number of Go cams. And what I do is, I can just go in here and I can start typing. So I just started typing Patrick Hassan, chose him.
Yeah.
So that's really these are the ones that are going to be the ones you're interested in.
Is the filter by term? What is considered a term?
Yeah, we can try it. We can try filtering by Go term. So,
So those are Go term. Okay.
Yeah, so I can try immune response. But I will tell you, I haven't tested this functionality, so let's find out. Let's see how it does. Yeah, so okay.
Yeah, so it has viral responses. It has
Yeah.
various.
That seems to work okay.
It's a pretty broad term.
Yeah, exactly. Yeah, and these are all Go terms. So what I hope it's doing, and it looks like it's doing, is taking advantage of the ontology structure to also recognize more specific terms.
Can you find pathways in specific cell types?
We can. So we can search. So in the term, we should be able to search for cell types. If they're annotated, so because as I showed you, the cell type field is legal in GoCam, so you can actually enter cell types. In practice, I have no idea how heavily it's been used. So we can try. You want to try a cell type? Let's take a look. I wouldn't count on it. Let's give it a shot. You have a cell type in mind?
Sure. T helper 1 cell, CD4 positive T helper 1 cell.
Okay, so I go T dash helper.
That's a question. Could be
Or T cell.
Or just
so
TH1.
Or TH.
Try TH.
TH1.
TH1.
Yeah, so this is going to search the cell TH1 like that.
Yeah, so I didn't use that.
Yeah,
Okay.
there you go. Okay.
I've got processes in those. Yeah, there we go. TC1 cell? No, not the same thing.
I don't think that's.
There we go. This, no.
Interesting one.
There we go. Yeah, try that one. T helper 17 cell. Yeah.
Okay, let's give it a shot. Okay, well, what do you know? Okay, yeah, there you go. I guess so again, but I think, a lot of these are going to be very simple models where it's just got a single statement. But anyway, yes, you can search for any ontology term there, and that's the main point. But yes, this is something where I don't know how complete. So our usual curator guidance is, don't specify the cell type unless this process is really highly specific, or this part of the process is highly specific to that cell type. So as I said, I haven't reviewed how extensively we use that, but this could also be another thing where if you notice that information is missing from our models, that's another thing you can provide feedback on. So, again, or, as I said, if you have curators who would be interested in working directly on models, that's something we could talk about as well. But the easiest way to get us to make changes is just to put a simple request in the GitHub repository, or send an email to the Go help desk. So all of this, Go is really very community oriented, and so we respond to requests for updates really very quickly.
The experience that I was having, Tom, is that if you look for process So I put like apoptotic, for example, then it gives a bunch of different cell types, you
Oh.
know. So process might be the best way of looking for it. I don't know.
Yeah, but that's really nice to see. This is always my fear of doing these live demos. You know, people ask you for something and you're wondering, does it really work as I hope it works? So this was, I'm happy to see this.
Yeah, thank you again. So we have 5
my pleasure.
minutes. Any other questions? I would love to see the real-time collaborate feature, but that might be difficult just with you only in there, unless you would probably have to log in from 2 different browsers. That might be too difficult. So
Yeah, that's right.
Yeah.
Yeah. But you know, as I said, we can always schedule a follow-up if it turns out to be a useful thing to do. All right.
Well, if we don't have any more questions, we can conclude. Thank you so much again for this wonderful presentation. Really appreciate it.
Yeah, thanks.
Too simplistic question, maybe.
Yeah.
Sometimes in presentations in immunology seminar or so, I see a very, very complicated diagram, and then the speaker focusing on only small part of that diagram, and then this is the pathway that is the focus of the talk.
Right.
So my naive question would be, if I'm not a biologist trained, and I'm not an artist in that sense, if I see this complicated scheme, can your system help me to identify the partial small part of the complicated picture to focus on? And it may have been overlooked by some other people.
So yeah, that's an interest. I mean, I think I understand what you're getting at, but I think what is interesting to one person may not be interesting to another. So how do you know? So I guess, you know, my answer would be, what you can see here is that you can search for things that you know, so you could search for a gene product. You could search for a function or a term, in this case, a sort of cell type that you're interested in, highlight things like that on the model. I guess, is that what you're talking about, or is that not what you're talking about? Like how oh, I guess then another thing we try to do is, as I say, we nest biological processes, so you should be able to focus on a sub process within a larger process. But yeah, these are really good questions, because I think this is something that everyone struggles with. I guess my sort of natural instinct is to ask a question back and to understand a little more. What are the sorts of things that this group is interested in, and how can we help with that? I think is probably, a useful question to ask. And I don't know if now is the right time, or if we wait until later on. I guess you said there was going to be a discussion starting in a couple of minutes. Is it better to wait until later? But if you can give me a little more idea about, so what was your, what were you interested in when you wanted me to give you a little introduction to GoCams? And is there something we can explore around what we can do to make this as useful to you as possible?
Well, if I'm trying to understand the art of a speaker who is a specialist, okay? And not necessarily to copy them or mimic them, but to follow their intuition, how they were able to focus on a partial picture of a very complex picture. And maybe there is another attractive partial picture that they overlooked. And it looks to me, I'm not an expert at all, but it looks to me that the tool you are describing is very powerful in this direction.
Yeah, it's interesting. I'll tell you, I hadn't really thought about, what we could do to underline, to think about, what stories do you tell from a particular pathway or part of a pathway. What our aim really has been to basically have an infrastructure where you can have a community project integrating information from lots of different sources, lots of different publications into sort of a comprehensive model of a system. And that's really what we've aimed to be able to do here. One of the things, and as you say, there are lots of ways one might explore such a diagram. You can ask, what were the, because we have an evidence trail on everything. You can say, all right, I've got this publication. What were the pieces that this publication added to a model? So you can even answer questions like that. So there, and in a sense that ties to at least the stories that someone wrote down in the literature. But how you focus in on particular places, what we found with other, with just Go annotations in general is it's really useful when you have a corpus of knowledge, when you have statements that could be conflicting with each other, for example, right? So you'll have, one annotation saying that a gene product carries out a particular function, another one with a different publication saying it doesn't carry out that particular, that particular function. And sometimes these point out genuine discrepancies in the literature. And sometimes genuine debates that are currently going on in the literature. But more often they actually reflect context. So it's actually context dependent. And so those models haven't yet said, well, here's how it works in a T helper 17 cell, but there's another cell type where actually the system works differently. And another gene product isn't expressed. And so again, we don't need to have we don't have the requirement that a model has to be completely self-consistent. And so a post hoc analysis of a model might be really interesting way to look for at least potential discrepancies in the model. And so other things that someone might want to look at, or another way in which the same process was modeled differently by a different group using different evidence.
Well, you are focused on gene products, but there is a broader view to understand pathways. And your tools seem to be very powerful in terms of pathways.
Yeah, no, that's right. Yeah, I mean the gene products and their activities are the building blocks for the pathways. Yeah, it's exactly the way we try to model it. Mm-hmm.
Okay, thank you.
Yeah, the part of the complexity though is that an animal only has one pathway. Right. There is only, every pathway interacts with every other pathway. So you're always artificially carving out some sub pathway, until you build a model of everything. If nothing else, I mean, all the pathways are competing for the same energy resources. So they all absolutely are interacting with each other at multiple levels.
That's right.
The user accounts for the Gene Ontology or via GitHub?
Oh, you mean the user accounts for this?
Yeah.
Yeah, that's right. Yeah, no. So no, the user accounts actually are via ORCID. So, or yeah, so you'll see each contributor. So if we put in me, for example, right, you'll see my ORCID here.
Yeah, I'm
Right.
just wondering how to log in because I'm in an endless loop trying to log in from the Noctua page.
Oh, sorry. Yeah. So from here, you're not going to be able to log in because so what we do is, there's an application process to get curation access. So you can view everything. You're just not going to be able to. So you have read access. You just
Okay,
aren't gonna be
so
able
if you scroll up to the top of the page, there's a
Yeah.
login button and it doesn't go anywhere. And a quick search doesn't show me any way to sign up, to create an account, to make an application, so if you log out, if you disconnect your account.
So go up here to help.
Yeah.
Okay. And there should be instructions in here for how to get an account. User account setup.
Okay. Usually that would be linked to the login button.
Okay. Yeah, that's a good suggestion, actually. I like that.
Because you know what? That's actually quite difficult to find.
Yeah. Yeah, so help wasn't obvious. Do we need to put a big flashing sign somewhere down?
No, when you click on the login button, one of the options under the login button is generally create an account,
Yeah, that's good.
right?
Yeah.
If I want to make a new Gmail account, I don't have to go to Gmail help to figure out how to do it.
Yeah.
Because the document is probably 500 pages long. Okay.
Yep. Yeah, so I guess that really would be my question. Do you have, are you or others there interested in actually curating pathways? Are you interested in modifying existing pathways through the curators we have? Are you just interested in downloading and using the pathways? I mean, I guess I'd love to hear sort of how you envisage, what would you find useful for your project?
Well, we're a very heterogeneous group, so
Yeah. So I'd answer that. I'd ask that question broadly to everyone.
Yeah. I think Thomas might be interested in using some of the GoCams as part of his immune digital twin. Be one approach of making standardized shareable, annotatable, defensible models or model representations that are really not models. I think other people, no doubt, would have quite different uses. So does anybody else, let's see, it's a little after 4. Any other questions? We still have a few people on. I know Thomas had to drop off. He had to go pick up his son late in the afternoon and time to pick up the kids from school. So if there aren't any more questions, I thank Dr. Thomas. Thank you, Paul, very much. Very interesting. Go has been growing over the decades and just getting, more and more complete and having more and more functions added into it. Okay, one last really quick question. What about searching on Kebi molecules? I didn't see that in the search page. And a Kebi wouldn't be a gene ontology term, so it wouldn't be in the term list.
Yeah, so as I showed you, the term that we actually search for, T helper cell, is or whatever, T helper cell is not a Go term either. So we actually, that brought up a term from the cell ontology. So all the ontologies that Go uses, that GoCam uses, are searchable from that same place. So yeah, you can search for a Kebi term also.
Okay, so that's a very generic search box, which is really handy. You
Correct.
don't necessarily need to know what kind of thing it is you're looking for. Okay.
Yeah, yeah. And as I said, it's really designed, to help curators figure out if there's already a model of something. And so they may not want to build something from scratch. And so instead they can, either talk to another curator or extend a model, or, we also have a model copy functionality where you can copy a model and make changes to it. So, all this sort of stuff is in the tool. And we have a user facing site that, as I said, is, we're refactoring it right now, and it's clunkier than this. So this is the best I can do for you for now.
No, I was just trying a few things.
Yeah.
I noticed so that it's actually a quite powerful search box, because if I type in acetaminophen, it responds with paracetamol. So the drug has two names. And
Yeah,
that's
exactly.
it's really
I know.
handy that it's not only doing the term, it's also doing term synonyms,
Yeah,
which is
yeah,
really, really slick. That's really handy.
yeah, it's one of the nice things about operating on these ontologies, because you can use all the synonyms, and you can even, widen searches to use definitions. And
Yeah, yep. Of course, then you start running into two genes with the same synonym.
yeah, that's right. There's a point at which, you have so many options to scroll through.
Yeah.
So yeah, that's always the fine balance.